Today in this podcast, we're going to be talking about how DNA is the instructions for making a protein. We are focusing on section 12.3 in your book, in which we will be able to explain the difference between DNA and RNA, the difference between messenger RNA and transfer RNA, the process of transcription and translation, and how all of this combines to be able to make a protein. So we're focusing on section 12.3 in your book. First of all, we know that all cells undergo replication. So they make exact copies of each other, which is what we call replication. And this occurs in the nucleus with the DNA being the instructions for this. So the DNA has to copy itself. We know that DNA contains sugar, deoxyribose, the phosphates, the nucleobases, in which cytosine always pairs with guanine, thymine always pairs with adenine. But this is contained within the nucleus for exact replication of the cells. Now, what we're trying to do here now is to get these directions, because that's what DNA is inside the nucleus, is the directions for making protein. It's the code of life. And somehow we have to get those instructions to the ribosomes where the protein is manufactured. But DNA can't leave the nucleus. So therefore, the cell has to get the instructions from the nucleus to the ribosomes, and it does this using RNA. Now, RNA is a little bit different than DNA, and the three major differences of RNA is that instead of thymine, it uses uracil. So A bonds with T, so adenine bonds with uracil, not thymine. It's a single strand, not a double strand. And instead of having the sugar deoxyribose, it has the sugar ribose. And so this RNA is going to take the message from the nucleus, the instructions, and deliver them to the ribosomes so that the ribosomes can make the protein. The first step in this process is what we call transcription. And in the process of transcription, RNA makes a copy of the DNA. C still pairs with G, but now A is going to be pairing with U. So this is the first thing that happens. So essentially, the double helix of the DNA sort of unzips, allowing a single strand to be made from the DNA. So a single strand of RNA will get made from the DNA. This, at this time, is going to be called messenger RNA, because it's going to take the message, the instructions, from the nucleus to the ribosomes. So at this point in time, the cell is undergoing transcription, and then the messenger RNA will find its way to the ribosomes, where it will undergo the process of translation, where this RNA gets translated into proteins. I like to say it like this. If the nucleus is one country and the ribosomes are another country, and they don't speak the same language, one speaks Japanese, one speaks French, the information has got to be translated. So the RNA has to get translated into a different language inside the ribosomes so that it can be understood. So you've got translation taking place. It's now called ribosomal RNA. So the two major processes will be transcription and translation. So during transcription, the DNA is a template for the construction of RNA. It's called the messenger RNA, and it's going to take the message from the nucleus to the ribosomes. And that's because the DNA can't leave the nucleus. So somehow the instructions have to get from the nucleus to the ribosomes, and they're carried there by messenger RNA, mRNA. So the, as you see here, kind of a picture of transcription. Inside the cell here, inside the nucleus, you've got the double helix DNA strand that unzipped momentarily so that a single strand of RNA could be made inside of the nucleus, and then the RNA will break free and travel to the ribosome, and the DNA will rezip itself. Then, inside the ribosome, the messenger RNA is going to be translated into tRNA, which is also known as transfer RNA. Again, the ribosomes speak a different language than the nucleus. The messenger RNA gets read 3 bases at a time, which is what is called a codon. And there is a stop codon so that the ribosomes know when to start building the protein. So the 3 letters are a codon, and they code for a specific transfer RNA. And this transfer RNA is going to bring in the amino acid. There are 20 amino acids found in all species. So this here is what you see is the transfer RNA. On one end of the transfer RNA is an anticodon. On the other end is the amino acid. So the ribosome is reading the codon of the messenger RNA, and that tells which transfer RNA to bring in, which has an anticodon on one end and an amino acid on the other. So if you look here, AUG is the initiation codon. So the ribosome is reading, it sees AUG. That is a signal to the ribosome to start building the protein, bringing in amino acids. So here you have the transfer RNA. It's got the anticodon here, transfer RNA, and the amino acid here. Okay. So essentially you've got this ribosome reading the messenger RNA. It sees AUG, which is the start codon. That signifies an incoming transfer RNA with an anticodon on one side and an amino acid on the other. And it will continue, the ribosome continues to go along and read 3 letters at a time, read the codons, signifying the transfer RNA to bring in an anticodon and an amino acid, and it continues to build this chain of amino acids into a protein. Now there are also stop codons, and in your book you have a chart of all these codons and anticodons in chapter 12 on page 338. So it's going to suddenly read a termination codon or a stop codon, one of the 3, and that signifies the end. So this protein stops being built here, it stops bringing in amino acids, and you have created this string of amino acids called a polypeptide, which will be folded then into a protein. So you've got the DNA strand, which gets transcribed into mRNA. And again, the difference for mRNA is that it's a single strand. It contains uracil instead of thymine, and it has the sugar ribose instead of deoxyribose. The messenger RNA takes the message to the ribosomes, and in the ribosomes it is translated into amino acid language, reading 3 codons at a time. It brings in the correct amino acid, puts the amino acids together in the correct order to make a protein. This picture here is just another drawing of that entire process. A little more information than what we're going to go into, but same general idea. The DNA unzips momentarily so that the messenger RNA can be created. The messenger RNA then travels to the ribosomes. In the ribosomes read the messenger RNA 3 letters at a time, which is a codon. That brings in a transfer RNA, which has an anticodon on one end and an amino acid on the other. It strings the amino acids together to make a protein, and that becomes the entire process of how a protein molecule is made. And then you could get into more detail. We're not going to go into that detail as far, but the string of amino acids will then be folded into a protein, and depending upon the way they're folded depends upon how the protein functions. That gets a little more in-depth, but we're not going to go quite that far, but just wanted to give you an idea of what that looks like once the amino acids start getting folded into the proteins. So at this point in time, you should be pretty clear on transcription and translation and the process. There are 64 different codons that code for 20 different amino acids within a species. There is one start codon, but there are 3 stop codons that the ribosomes can read. And again, the big differences between RNA and DNA. RNA is a single strand. It contains ribose, not deoxyribose, and it has uracil, not thymine. And at this point in time, if you have any more questions, please check with your instructor or read your book. And again, just a little bit more, you can find this chart in your book about messenger RNA carrying the message from the nucleus to the ribosomes. The ribosomal RNA is what it's called when the ribosome is reading the RNA. The transfer RNA brings in the amino acid as the messenger RNA gets read. It brings in the correct amino acid and strings them together.